Golgi apparatus will contain enzymes like pepsin. If it is a pancreatic cell, it will contain enzymes like pancreatic amylase, trypsin, chymotrypsin. So secretory cell will have Golgi apparatus predominant, and all those secretions will be stored in the Golgi. As and when required, Golgi apparatus will release those secretions in the lumen of the particular organ as such. So that is one important organelle. And there are other structures like cytoskeleton, which will give shape and size to a particular cell. Different cells are having different size and shapes depending upon where it is located and what function is attributed to it. So that cytoskeleton will be there. And some other less important organelles like some granules are there, which are concerned with certain secretions, certain proteins which are specific for that particular cell. As far as nucleus is concerned, again we will be just touching the important parts in the nucleus. Nucleus will have two ingredients, one will be DNA, another will be RNA. RNA will be present in the nucleolus, while remaining part will be made up of DNA, or in other words chromosomes. So DNA or chromosomes will be concerned with genetic character and of course control of so many enzymatic activities which are going on in the cell. That regulation will be done by that DNA component, chromosomes, while nucleolus, which is containing RNA, is concerned with protein synthesis. So these are the two ingredients of the nucleus, chromosomes and nucleolus, having different functions. Nuclear membrane or membrane of the mitochondria, membrane of the endoplasmic reticulum, or membrane of the Golgi apparatus, all membranes are almost the same except one or two differences. Almost all membranes are the same, including the cell membrane, and that's why these membranes we call them as unit membranes. So we will be learning the structure of the cell membrane, but same structure will be applicable for membrane of any other organelle also, with little variation. So now we go into the details of that. We call it as a unit membrane. Cell membrane is a unit membrane, or what structure we will be learning is a unit membrane structure. When I learned cell membrane, at that time the concept was two lipid layers, two protein layers sandwiched between the two, and at that time concept was it is a rigid cell membrane. But now we have gone ahead with different research, and now it is proved beyond doubt that cell membrane is fluid in nature. All the time it is flowing, and in that fluid membrane proteins are floating. So that concept or that structure of the cell membrane is called as a fluid mosaic model. It appears surprising, but that is a fact, that membrane is fluid in nature, flowing all the time, and in that proteins are floating. So in that fluid mosaic model of the cell membrane, we are having three ingredients. Lipid ingredient is maximum. It contributes almost 60 to 70% of the structure of the membrane, made up of phospholipids and cholesterol. Phospholipids, as the name is, it contains that phosphate group and a lipid component, fat component. Cholesterol, all of you are acquainted with. Remember, many times we blame cholesterol for so many diseases, but cholesterol is useful also to our body. All cell membranes, they contain one ingredient, and that is cholesterol. So one should not have, say, totally cholesterol free survival possible. Otherwise membrane will not be formed only. So little cholesterol is required. When levels go beyond a limit, then it becomes harmful. But otherwise every cell membrane has got a ingredient made up of cholesterol. So lipid part is phospholipid plus cholesterol. Protein part is made up of lipoproteins. Again, as the name indicates, these proteins are in combination with lipids, and that's why the name is lipoproteins. They are there. These lipoproteins in the cell membrane are further divided into, again, number of other proteins. Some are called integral proteins or carrier proteins. As the name indicates, they form the integral part of the cell membrane. That's why the name integral protein. Or another name for the same protein is carrier protein. Again, name itself is indicative of the function. They help in transportation of the substance. A substance will sit on that carrier protein, and then it will be taken either inside or outside. So integral or carrier proteins is one ingredient. Another type of the proteins are channel proteins. We will be learning those channels also in detail later on. These proteins, they form ionic channels in the cell membrane. These are like pores which are present in the membrane for movement of the ions like sodium, potassium, calcium or chloride ions. They pass through these channels, and whatever proteins are lining that channel, we call them as channel proteins. So that is another type of a lipoprotein present in the membrane. And third type of proteins are peripheral proteins. These peripheral proteins are attached to carrier proteins and on the inner side of the cell. That means these peripheral proteins are projected in the cytoplasm. So naturally, as it is projected in the cytoplasm, it will have enzyme-like function. So it is catalyzing different metabolic processes in the cell. So carrier proteins acting as a carrier for substances like glucose, amino acids, channel proteins for movement of the ions through those particular channels, and peripheral proteins which are acting like enzyme for catalyzing particular chemical reaction. These are the three types of proteins present in the membrane. Coming to the third ingredient, that is glycocalyx or carbohydrate part of the cell membrane. This is lipid, protein and carbohydrate. These are the three ingredients. This is the carbohydrate part, glycocalyx. Now this glycocalyx is present only on the outer side of the membrane. This is the important point to be kept in the mind, that glycocalyx can't be inside the cell membrane. It is always on the outer surface of the cell membrane, and it is in contact with the proteins, those integral proteins or channel proteins, what we have mentioned earlier. This carbohydrate part is attached to this protein moiety, and as it is projected outside, it will have easy access for the substances which are coming from outside the cell. For example, certain hormone which is coming through the blood, or certain drug which is coming through the blood acting on the cell membrane, that drug or that neurotransmitter hormone will be combining with this glycocalyx first, and then cellular function will be getting altered. So main role of this glycocalyx is it provides attachment for various hormones, drugs, neurotransmitters for the cellular function to get modified. Similarly, this glycocalyx also provides cell-to-cell attachment. It is said that adjacent cells are attached to each other and there has to be some cementing substance between the two cells. So this glycocalyx acts as a cementing substance, so that cell-to-cell contact is established, and that gives a particular shape, size to the tissue as such. So that is another role done by glycocalyx, that it provides attachment with the adjacent cell. So providing site for hormone or drug or neurotransmitter and cell-to-cell attachment, these are the two functions carried out by glycocalyx. Now we go into the details of that structure in form of a diagram. Here you can see phospholipids are maximum, or lipid component is maximum. Now in this lipid component you can see there is one hydrophilic portion, hydrophobic portion. Each lipid molecule has got two parts. Hydrophilic, the name itself indicates. Hydrophilic means it is having affinity towards fluid. So this globular part is hydrophilic and this tail-like part is hydrophobic. And these are the lipid molecules arranged in such a way that hydrophilic portion, one part will be directed outside